This presentation is about DNA. DNA is deoxyribonucleic acid, and it's found within the nucleus of the cell, so right here, and it's generally in the form of what we call a chromosome, like here. But the only time it's in a form of a chromosome is actually just before cell replication or cell division, meiosis or mitosis. Generally, the actual DNA is just a really long strand that we can't actually see under a microscope. It's only just before that cell division that we see it when it coils up into the form of a chromosome. So as you can see here, it's all coiled up into the form of a chromosome, and it's actually histone molecules there that hold that coiling into that chromosome shape. The DNA has a very interesting sequence in that it's actually two molecules, one molecule on this side here and the other molecule on the other side, and they're actually held together through hydrogen bonding, and that hydrogen bonding gives the structure of what we call a twisted ladder, or specifically scientifically we call that a double helix. So the double helix shape, which is like the spiral ladder. If we look a little bit more closely at the DNA, we can see here in this particular picture here the hydrogen bonding. So the hydrogen bonding between the base pairs, and that's really important that we understand that as the base pairing rule. With this base pairing, every adenine will pair with a thymine, and every guanine will pair with a cytosine. Why did I draw three lines between the guanine and cytosine? That's because there's three hydrogen bonds between that grouping, whereas there's only two between the adenine and the thymine. And that's for later on, for year 11 or 12 chemistry or biology. So if we look again at this structure, we can also see the backbone. The backbone here is made up of two different molecules, the ribose sugar here and the phosphate group just here. Both the sugar and the phosphate are the backbone, and they hold individual bases. So the actual sugar itself is the section that holds the base, and a sugar, a phosphate and a base, so like in this group here, or particularly probably easier to see if we look at this group here, this is what we call a nucleotide. And essentially, the DNA is actually made up of nucleotides all joined together in a long row, and then that long row connecting through the hydrogen bonds to the alternative strand. Now, you can often get asked to potentially work out the complementary strand. So let's say we had one strand that had the letters A, T, G, C, G, G, T, A. And if you were needing to make the complementary strand to that, then obviously you'll need to write the matching base pairs. So for this one, if we're looking at A, the complementary strand would have T, and the next letter T, its corresponding base is A. G goes with C, C goes with G, and then C, C, A, and finally T. So that would be our complementary strand to the original strand. Now, sometimes when making that complementary strand in the process of DNA replication, now DNA replication happens for two reasons. One reason could be that the cell needs to go through cell division, so mitosis or meiosis, in which case the DNA will replicate and make another DNA strand. Or the other process that DNA replicates is actually to make a copy for RNA. So the copy is called RNA, and that RNA can actually get out of the nucleus and be able to be translated into amino acids and a protein chain. So if we have a look at the process of replication, we can see that sometimes it doesn't quite go correctly. So generally, we see that the DNA before, we've got nice complementary base pairing with G with C and the T with the A. And ideally what will happen is that we have a break off and we have the complementary bases coming in. So that the correctly replicated DNA should look exactly like the original strand. However, we can have replication errors, which are what called mutations, and mutations occur when we have a mismatching pair. So in this case, we've got the thionine, which is supposed to be matched with the guanine, and in that instance, you won't end up with hydrogen bonding between those two. So the DNA actually doesn't fit nicely together. Not a good thing, obviously, when making the cell, and hopefully during that mitosis or meiosis process, the error will be picked up and the cell will be basically said, no, time for destruction, this is not quite right. But obviously mutations do occur, and they may give rise to new alleles or new characteristics for that particular gene. So this is the mutation.